I'm Ted Schutzbank, the Senior Manager of Scientific Affairs for Infectious Diseases at Agena Bioscience. Today, we're going to discuss the need for SARS-CoV-2 targeted variant testing and how Agena Bioscience has met that need through the development of the MassARRAY SARS-CoV-2 variant panel. Mutations in the SARS-CoV-2 genome is a byproduct of viral replication, especially as selective pressure is put onto the virus by the host immune response, either through natural infection or by vaccination. The rate of genetic variation of the SARS-CoV-2 virus is less than for many other RNA genome viruses, since SARS-CoV-2 has a proofreading function that corrects errors in the nucleotide sequence that may occur during RNA replication. But as we're currently observing, such errors still do occur and can lead to variants with increased evolutionary fitness. The emergence of the SARS-CoV-2 variants that are now circulating in the US and worldwide have essentially replaced the founder Wuhan 2019 lineage due to increased fitness in the areas of virus transmissibility, virus replication, and escape from immunity. It has been noted that one of the collateral effects of genetic variation in the SARS-CoV-2 virus genome is on diagnostic tests using nucleic acid amplification technologies, or NAT. False negative results due to nucleotide variants appearing in either the primer or probe target sequence of PCR-based assays have been reported. Thus, it is important that all tests have multiple virus gene targets, so that if one target sequence is so affected, a positive result for that genetically altered virus could still be achieved. Like any laboratory test, the results must always be considered in light of the clinical picture of the patient being tested. This is precisely why Agena's EUA assay, the MassARRAY SARS-CoV-2 panel, was designed as a multiplex with 5 unique targets within the genome, providing resilience against mutations. This is the same technology with the high multiplexing capabilities used to create our SARS-CoV-2 variant panel. So why is strain surveillance for SARS-CoV-2 necessary? There are multiple reasons which are pointed out on this slide. The different variations now in circulation have gene alterations specifically in the virus spike glycoprotein that can have significant effect on the health of the populations in which they have been observed. These include the ability to spread more quickly from person to person, increased pathogenicity, the ability to evade the host immune system in both unvaccinated and vaccinated individuals, and decreased susceptibility to monoclonal antibody therapy. Because of these factors, knowing which variants are circulating in the community and possibly knowing which variant an individual is infected with may yield critical information for the clinical management of infected individuals. The Centers for Disease Control, the CDC, has stratified the classification of recognized SARS-CoV-2 variants currently in circulation. Variants of interest are those that have been reported to have increased transmissibility, possible immune escape, possible resistance to therapeutics, or escape detection by diagnostic testing. These VOIs may cause an increased proportion of cases in a specific population, but have limited prevalence in the overall population in the US and in other countries. The variants classified as VOIs are shown on this slide. I won't discuss all of these individually, but this table can be found on the CDC website shown on the slide. There are currently 4 such SARS-CoV-2 variants, one of which, B1526.1, was just added in the past few weeks. 3 of the 4 CDC VOIs are variants first identified in New York State, with the 4th being a variant identified in Brazil. SARS-CoV-2 variants classified by the CDC as variants of concern are those that have been determined to have a more significant impact on public health, with evidence that they negatively affect susceptibility to one or more classes of therapies, decrease neutralization by antibodies elicited through infection or by vaccination, or have a negative impact on diagnostic procedures causing false negative patient results. The VOCs shown on this slide have been extensively covered in the scientific literature as well as in the public press, and include the UK, Brazilian, and South African variants, B117, P1, and B1351 respectively, as well as two variants that have been identified in California, B1427 and B1429. The last of the CDC variant classifications is variants of high consequence. These are variants that show clear evidence that prevention measures or medical countermeasures have significantly reduced effectiveness when compared to previously circulating variants. The emergence of VHCs in the community would force public health officials to declare a public health emergency of international concern. None of the variants identified so far fit into this classification. It is in the event of the emergence of a VHC that variant detection will have the greatest utility and public health benefit. And if all instances of a VHC can be identified and effectively contained. As we have seen with the COVID pandemic as a whole, and the emergence of current VOIs, VOCs specifically, these variants spread. The time is now to develop infrastructure and capability to combat the real possibility of the emergence of a variant of high consequence, as responding and building this capability after detection will be too late to have a containment effect. Let's turn our attention now to the identification of SARS-CoV-2 variants. As I discussed earlier, genetic variations in the SARS-CoV-2 genome can lead to false negative results for certain nucleic acid amplification-based tests. One commercial test in particular specifically targeted the spike gene. Fortuitously, the delta H69 delta V70 mutation in the spike gene caused a total loss of signal in this PCR assay. However, in some cases, only reduction of signal or late amplification was observed. This phenomenon has been referred to as S-gene target failure or S-gene dropout. It was suggested both anecdotally and in some publications that such a result could be used to putatively identify the presence of the SARS-CoV-2 variant B117, which is a variant of concern. While the B117 variant does carry this mutation, it is also present in other variants, as evidenced by the results of the two studies shown on this slide. One study demonstrated that only 45% of the samples with S-gene dropout were confirmed as B117 by sequencing. The second study found a much lower percentage of samples with S-gene dropout confirming as B117 after sequencing. The various SARS-CoV-2 variants are each identified by their repertoire of gene variations. So the take-home message here is that one cannot call a variant by the presence of one mutation only. In addition, S-gene dropout could also be caused by other genetic variations in the targeted primer or probe sequences. And a false negative result is not necessarily indicative of one sequence, of one specific sequence mutation. Targeted variant screening tests are essential to be able to rapidly and economically determine what SARS-CoV-2 variants are present in a specific population. For such a test to be effective, requirements must be met which are listed on this slide. The test must be able to detect all of the currently recognized VOCs and VOIs. Such a test must be flexible enough to incorporate new variants as they are identified and emerge in the population. The test must be able to provide high throughput, that is, the availability to analyze a large number of samples per run, and provide results in a timely manner. Result reporting must be integrated into the software of the instrument performing the test without the need for extensive informatic analysis. The cost of testing should allow the economic feasibility of testing large numbers of samples. For the remainder of this presentation, I hope to convince you that the Agena Bioscience MassARRAY SARS-CoV-2 panel, Varian panel, meets all of these criteria. So for the remainder of this presentation, we will discuss how the MassARRAY SARS-CoV-2 Varian panel is a highly multiplexed assay, which allows the identification of SARS-CoV-2 variants in a single test. It is very cost-effective with integrated software that clearly interprets the generated data and presents results in a clear and straightforward report. Depending on the laboratory workflow, over 1,000 samples can be analyzed in a 24-hour period with a single platform. The instrument as shown here has two components, the CPM module on the right and the MALDI-TOF module on the left. The heart of the MassARRAY technology is the SpectroChip array. This is available in two variations, allowing for the testing of either 96 or 384 reactions per test run. The Spectra chip is assay agnostic in that all assays performed on the MassARRAY are performed using the same chip. The MassARRAY has a simple and straightforward workflow. After extraction of the sample nucleic acid, in this case the viral RNA, the targeted genome sequences are amplified by reverse transcriptase PCR using any standard thermocycler. After amplification, each reaction is treated with shrimp alkaline phosphatase to essentially remove any remaining deoxyribonucleotide triphosphates from the reaction. Lastly, an extension probe cocktail and the required associated reagents are added to each sap-treated reaction and subjected to an appropriate number of thermocycling events in the thermocycle. At the end of this process, the entire plate is loaded onto the MassARRAY CPM module for final processing and sample analysis. The CPM module automatically desalts each reaction, transfers a small set portion of each reaction to a single pad on the Spectra chip, and then transfers the chip to the multi-top spectrometer for analysis. I should mention here that the CPM module can process two 96 or 384 microwell plates simultaneously. Unlike most other NAT methods, MassARRAY technology is not dependent on fluorescent or colorimetric detection. Each of the extension probes that I just referred to has a known nucleotide sequence and a known molecular weight. During the extension process, each of the extension probes is elongated by a single nucleotide only, altering its molecular weight by the amount specific to which nucleotide was added. Once in the spectrometer, each pad is irradiated by the internal laser, which ionizes the extension probes on the chip, causing them to migrate upwards through the instrument's vacuum column. The rate at which this migration occurs depends on the molecular weight of each molecule, with those with lower mass traveling faster than those with higher mass. As they reach the top of the vacuum column, the ionized molecules impact the detector, and a signal is generated that is proportional to the number of molecules of each different molecular weight. The results are plotted as shown on this slide as clear and easily interpretable peaks, which are analyzed by the software to produce a clear and unambiguous result that we will shortly see. Now let's turn our attention to the MassARRAY SARS-CoV-2 panel itself. The multiplex primers and probes are split into two wells as shown on this slide. Shown on this slide is version 1 of the SARS-CoV-2 variant panel that was released in mid-March of this year. This assay detects 20 different genetic markers that define each of the original CDC variants of concern, B117, B1351, and P1, and additionally the MINC5 cluster, which was of interest to European laboratories. Also included is the G614G mutation, which is present in nearly all SARS-CoV-2 variants in the United States, and is concerned since it has been shown to have increased efficiency for infecting upper airway epithelial cells and replicates in greater numbers than the ancestral virus. Two controls are also included, one which detects the conserved N gene consensus sequence for the SARS-CoV-2, and another that targets the host cell GAPDH gene. Since the introduction of this assay, the CDC has identified additional variants of interest and variants of concern. As a result, we have designed and built version 2 of the variant panel shown on this slide. Variant 2 expands detection to include two of the New York variants of interest, B1526 and B1525. Also added in this version are the two variants of concern that arose in California, B1427 and B1429. By request, we also included a relatively new variant that emerged in Florida, defined by the spike protein alterations K1191N and Q439K. Two standalone genetic variants are also included, D614G, which is a carryover from version 1, and Q677P, which is found broadly across the United States and may enhance SARS-CoV-2 infectivity or transmission, akin to what was observed for D614G. It should be noted that the time interval between the release of version 1 and the availability of version 2 was less than 2 months, demonstrating the ability to rapidly update the MassARRAY CoV-2 variant assay to include newly recognized variants of interest and variants of concern. Agena remains committed to adding new variants as they emerge. The next slide shows the variant calling algorithm used by the software to call the variants based on the specific genetic markers detected in the patient sample. This algorithm can be modified by the user to meet the needs and requirements of the laboratory when requested. There are several ways the software can report test results. This slide shows one example, which is generated as a CSV file presenting the sample identification information, whether the test for each sample passed the necessary quality controls, the variant call for each sample, and a list of the genetic markers detected for that variant. This output can easily be exported to the laboratory LIMS system. Alternatively, a report can be generated in a PDF for each individual sample tested. This report shows the sample ID, the QC information for that sample, a list of each of the genetic markers detected, and the variant call. During this presentation, I have mentioned several times that the MassARRAY platform allows for high-throughput testing. The chart on this slide shows that just over 1,500 patient samples can be processed in an 8-hour shift with the single MassARRAY 384-well platform. This is significantly higher than what can be achieved by next-generation sequencing methods and requires no bioinformatics, as it is coupled with automated reporting software, allowing for high-throughput screening of many patient samples in a single day. This concludes our presentation on the MassARRAY SARS-CoV-2 variant panel. During this session, we discussed the need for a highly multiplexed SARS-CoV-2 screening assay to be able to follow the spread of variants of interest and variants of concern throughout communities and more broadly across the US and globally. We also clearly demonstrated in this discussion that the Agena Biosciences SARS-CoV-2 variant panel meets the requirements for rapid, cost-effective, high-throughput screening of known SARS-CoV-2 positive clinical samples through its highly multiplex design, the rapid turnaround time to obtain results, its high sample throughput, and the elimination of the need for extensive informatic analysis to report results. Agena has already demonstrated our commitment and capability to update the panel to include new variants of interest and concern in real time as they emerge. Without being an alarmist, realistically, now is the time to build variant detection infrastructure before the real possibility of emergence of a variant or variants of high consequence eventuates. To learn more about the MassARRAY CoV-2 variant panel, you can visit our website as shown on this slide. We are very committed to ensuring that this panel remains current as new variants of interest and concern emerge in the population. We ask you to help us in our surveillance efforts. You can propose additions to this test by emailing us at scientificaffairs@agenobio.com. I thank you very much for your kind attention to this presentation. Thank you.